Hi, my name is Jillian Calero, and the date is July 9th, 2020. Right then. Okay, awesome. Let's get started. So for this assignment, I chose the topic of regulation of the gastric phase in digestion. So digestion, the first two phases include the cephalic phase and the gastric phase. The cephalic phase is the very beginning of digestion. The cephalic phase begins with the sight, smell, and taste of food. It even can begin with a thought of food. When we think about food or smell food, we salivate, and our saliva contains acids that begin the process of digestion. The cephalic phase can also be referred to as a long vagal reflex. And then we have the gastric phase. So the gastric phase refers to motility and secretion of various hormones to continue the process of digestion. The gastric phase includes the distension of the stomach, which essentially begins the gastric phase. The gastric phase also includes activation of endocrine cells and enteric neurons via peptides and amino acids. So on this slide, I chose a diagram from Silverthorne. I really like the diagrams from Silverthorne. I think they do a good job showing what's going on in the different systems. So in this picture, we can see both the cephalic phase and the gastric phase and what's happening during those two phases. So let's follow the lines for the cephalic phase. First, we see that this person is seeing, smelling, and potentially tasting food, which indicates to the brain that food is available, and the medulla oblongata sends a signal to the preganglionic parasympathetic neuron in the vagal nerve. So then we have long vagal reflux. And then it goes to the enteric plexus, and then we see both a short and a long vagal reflex. This then moves to the postganglionic parasympathetic and intrinsic enteric neurons, which innervates the target cells, and then there is secretion and motility of the food that we are eating. Moving to the gastric phase, which is the short reflex, which is shown by the reddish-pink arrows. The short reflexes are initiated by distension or peptides and amino acids. So when food is introduced to the stomach, the food volume increases the distension of the stomach. The food can also contain peptides and amino acids that was initially broken down in the mouth. So then there's sensory input in the gastric mucosa, and then it makes its way to the enteric plexus, and then the postganglionic parasympathetic and intrinsic enteric neurons, which innervate the target cells, and that also in turn creates secretion and motility of the different acids in the stomach. So then in the last diagram, we saw the long vagal reflexes and the short vagal reflexes. We're going to go more into the short reflexes of the gastric phase here. So I chose a picture that I feel like does a really good job showing where certain cells are in the stomach, what they do, and what their job is in terms of digestion and secretion. For this slide, we're going to focus on the G cells right here. So these G cells secrete gastrin, and gastrin is released and is stimulated due to the presence of amino acids and peptides in the stomach, as well as stomach distension due to food volume. So the gastrin release can also be triggered by neural reflexes, which is mediated via gastrin-releasing peptide. Its primary function here is to release acid by acting on parietal cells as well as releasing histamines. So in the diagram, we can see very easily that the G cells are producing gastrin, which in turn is influencing ECL cells, which is in turn producing histamine, and is creating a positive feedback loop here. So there are different secretions in the gastric phase of digestion. I simply typed them out here, but I'm going to talk about them in more detail as we go through them. So the first secretion in the gastric phase is gastric acid. So the gastric acid is secreted by the parietal cells. The gastric acid's function is to activate pepsin and to kill bacteria. The gastric acid is stimulated by the production and presence of gastrin as well as histamine, and that just goes back to the positive feedback loop that we saw in the last diagram. And then we have pepsin, which is an enzyme. So pepsin, or pepsinogen, is secreted by chief cells. So the function of pepsin is to digest proteins, and pepsin is stimulated by the presence of acid. The next secretion is gastric lipase, which is also an enzyme. Gastric lipase, just like pepsin, is secreted by chief cells, and the function of gastric lipase is to digest fats. Gastric lipase is also stimulated by the presence of acid. Then we have histamine, which is a paracrine secretion via the gastric mucosa. So histamine stimulates gastric acid secretion, which goes back to that positive feedback loop that exists when the G cells are secreting gastrin. And that explains why gastrin stimulates the production of histamine. So then we have somatostatin, which is also a paracrine secretion. So somatostatin is secreted by D cells, and it inhibits gastric acid secretion. So, referring back to the last diagram, there's a positive feedback loop that exists with the G cells and the production of gastrin and histamine, but when somatostatin is present, it creates a negative feedback loop. The somatostatin stops and inhibits the production of these acids that and essentially will stop digestion. And then we have intrinsic factor. An intrinsic factor is secreted by the parietal cells. Intrinsic factor complexes with vitamin B12 to permit absorption. And intrinsic factor is stimulated by the presence of gastrin and histamine. So then we can move on to the initiation and integration in the gastric phase. So this is another Silverthorne diagram. It takes you through both the cephalic phase and the gastric phase once again. It goes into a bit more detail than the last diagram did from Silverthorne. And it's a little complex, so I broke it down a little bit more in the next slide. So the diagram is essentially showing us that the cephalic phase begins once food is introduced. Whether we see it, smell it, taste it, the cephalic phase is beginning. When food enters our stomach, the stomach is distended and the gastric phase begins. The gastric phase is controlled by distension of the stomach, just like I said. The distension of the stomach is an indication that we need to start releasing gastrin. That gastrin stimulates acid production, which in turn stimulates pepsinogen. And a negative feedback loop occurs when somatostatin is released and decreases the production of acid and pepsins. So now we go into the balance between digestion and defense. So the main function of the stomach is digestion, But as we know, in the stomach there are very strong acids, and if there was no barrier between the stomach wall and the gastric acids, then you would destroy the lining of the stomach. So the stomach is producing acids in order to assist with the digestion of food. However, the stomach also has a defense component. So in the stomach, there's a mucosa bicarbonate barrier, which provides protection against autodigestion. So autodigestion is when we digest our stomach. So there's two components. The two components are laid out in the name itself. There's the mucus, which provides a physical barrier between the acid and the stomach lining, and then the bicarbonate creates a chemical buffer barrier. So a chemical buffer barrier means that since the acid is existing on the inside, we have the bicarbonate, which is a basic chemical, and that is counteracting one another. And then we have the mucus cells. The mucus cells are located on the luminal surface and on the neck of the gastric glands. The mucus cells have the abilities to secrete mucus and bicarbonate to create that mucosa bicarbonate barrier to avoid this autodigestion from occurring. And that's it. Thank you so much.